Good evening, ma'am.
Good evening, Shubhansh. Huria, have you joined with two devices? Huria, have you joined with two devices? Good evening, children. Now turn on the videos. Okay, so Abhinav, in the last class, what we have completed? So that what we did in the last class? Very good. Shubhansh, what we did in the last class?
We did vegetative propagation. No, we did.
About the bread, something we did.
Did we do yeast?
See, Pranay, I know I have to start vegetative propagation, but my question is what we did in the last class? Which topic we completed? Yeah, spore formation. There done on the video. Shubhansh done on the video.
Yes, ma'am. 1 minute, ma'am.
So last time we have completed with the spore formation. We discussed about the structure of the rhizopus and how the rhizopus they will be multiplying, they'll be dividing, and ultimately they'll be producing so many, the organism altogether. Fine. Now, before going to the vegetative propagation, we have two small topics to discuss with. So let us complete those two topics. The first one is the differences between fission and fragmentation. So, yeah, tell me, Pranav.
Ma'am, actually, tomorrow I have my exam, like science exam. So
Hmm.
I had a few doubts from life processes, ma'am. Like this one doubt from this one experiment.
Tell me.
Ma'am, it's like we have 1 second, ma'am. Ma'am, we have this like these leaves with patches, right, ma'am? Variegated leaves, like all leaves. There was this one experiment on that activity. So I had this doubt why we put these leaves in boiling water and then like put it in alcohol later.
To remove the color, to decolorize. Actually, the main word you will be using there, to decolorize. So as to remove the color of it. Okay.
Like, what's the point of removing the color too?
Ma'am, I'm trying to type my question on the.
See, if you have the green chlorophyll there, then what happened? The experiment will not be successful. So that is why what they have to do with the help of the alcohol, they will be decolorizing it so that exact and appropriate result we will be getting. Okay.
Okay, ma'am.
Then any other doubt anyone of you have? I think so many messages have
Ma'am,
come.
can I show it?
I want to listen to the recording division also. Aryan, it will be taking some time actually for the recordings and all, because the member is not present from. Yeah, I know the recordings are not there from the previous. I think almost some 10 days around something. So soon it will be. We will be providing you. Okay.
Okay, ma'am, because my exams have postponed, ma'am. So I wanted to listen once.
I know, but see, I can say that I need a day or two time actually, and 2 days I need for it. Okay, once it is available, I'll message you personally that it is available, and then you can go through it.
Okay.
Okay, yeah,
Can I
Abhinav,
show
tell
my
me.
question, ma'am?
Yeah, tell me. Yeah.
I will give you 4 scenarios in which we have to say that like lime water in every case, and we have to determine which is going to turn more milky.
Hmm. You're giving me the scenario in the chat, or I cannot see like that. Maybe you can. No, I cannot see set a. No, I cannot see properly. I think you have to share me the photograph on my number, Abhinav. Try and share it there so that I can explain you that point. And any other doubt, children, apart from Abhinav's doubt? Okay, till then, Abhinav is sending me. I'll go to the next differences now. Children, turn on the video. Kashvi, Meher, Hurya, Dhairya, Shubhansh. So we will be discussing before going to the vegetative propagation. The topic will be the differences between fission and fragmentation, and the fragmentation and regeneration. So sometimes what happens, they do ask for the 3 marks or 2 marks, the differences between the both, either the fission and fragmentation or the fragmentation and regeneration. So let's discuss the difference first of all, and then we will be going to the vegetative propagation.
Ma'am, if we write, I had
So
one board. Ma'am, if we write it in points, is it enough?
point, what you're saying, Abhinav, Aryan, can't get you.
Ma'am, in boards, if we write it in
Yeah.
point wise, is it like
Huh?
okay?
You should write only point wise in the board exam, Aryan. The paragraphs are not acceptable. If I am the evaluator, I will not be going through your paragraphs. I'll be just going to your, the points. That's it.
Okay, ma'am. And right, and if like content is long, but if it written in like short, like in point wise, although
That's
but
okay. That's okay, but the major keyword should be there. See, in the last exam, I think I have given you to explain the whole process of the filtration of urine. So in that case, if you are writing the main thing, reabsorption, secretion, second point, then ultrafiltration. So if these three main keywords are there and their definition is there, then definitely you will be getting the marks. But if you are not writing the name, which is reabsorption or secretion, then you will be getting half of it.
okay.
And I think that's the reason,
Ma'am,
for you all, I have reduced the marks, those who have not written the heading. Yeah, Abhinav, tell me.
ma'am, what if we write extra, like in the exam they asked me the structural unit of kidney and I wrote everything about kidney.
Depends upon your marks. See, they don't have time to see to check your whole thing and all for 1 marks. So they will be looking just directly to the nephron, and then just that's it. They will be leaving everything. They don't bother about it. That is why check to the mark. If your question, yeah, suppose they have asked you to write the structural unit of the kidney, then nephron. So they will be giving you the marks. This is suppose A. Now, second point, they have asked you that you need to write the, just 1 minute. You need to write the maybe 2 functions of kidneys. So what you can do, you have to write first of all like this, functions of kidney, then underline it. So this should be your heading, and then leave 1 line and then write your first point, then leave 1 line, then write your second point. That's it. So what they'll do, they'll see your point 1, they'll see point here they will give you half, here they will give you half. So like this, then and there they will be giving the marks. But same thing, if you write in the paragraph, sometimes what happen, 2 points you have mixed together and you have written and in between. They will think that is 1 point and they'll give you only half marks for it. So don't do that. If they have asked you 2 points, write it separately with the 1 line leaving between them. Okay.
Ma'am, see each point contains half mark.
Depends upon the question. It can be 1 marks also. Sometimes they ask you in the one question itself, they give you the three things. So it will be 1, 1, 1. So something we have called as a marking scheme actually. So in the marking scheme, what happened? I think we have. Just give me a minute. I'll just show you the marking scheme of one paper. Just a minute. I'll show you the marking scheme, what it is actually. Okay, so I'll show you the paper and the marking scheme. Is my screen visible, children, to you?
Yes, ma'am.
Okay, so this is the paper. No?
Ma'am, it's loading.
Ma'am, it's still loading.
Okay, let me stop sharing a bit. Now?
No, ma'am.
No, ma'am.
Manisha has started screen sharing.
Oh. I hope now it is visible, children.
Yes, ma'am.
Yes,
Okay,
ma'am.
so this is the paper of the year 2023, the sample paper released by the CBSE. So you can see, like in front of all the questions, they have given the marks also. Like question number 1, they've given the marks on the right hand side 1. Then 2, 3, 4, all they were like 1 marks. Now coming to the 21, 22, 23, 24, all are 2 marks. They have given only 2 marks, mentioned there 2 marks. Then 27, 28, 29, they were all like 3 marks. Okay. Then coming to question number 31, they have mentioned 1 plus 1 plus 1. Why they have mentioned 1 plus 1 plus 1? Because each and every part is of 1 marks. Then question number 32, they have given 2 plus 1 because 2 questions are there. The first question is of 2 marks and the second one is of 1 mark. So accordingly, then you should answer, and that is how the marking scheme will be. See, if I talk about this, see, in this question number 21, they have asked you something, though it was all together 2 marks, but they have given 1 plus 1 there. 1 for the first point, the second one for the second point. Same goes for the 22 question number. They have asked to write the 4 points. Now each point was of 0.5 marks. So 0.5 into 4, which gives you 2. So for every correct point, it was the 0.5 marks. So if you are writing each and everything in the paragraph, how many marks will be getting? Will be getting 0.5. That is why whatever the numbers they have asked you to write 1.2.3 points, accordingly you should do it. 23, they have given you the question there for 1 plus 1 actually. So this marking scheme, you are not aware. Only the person who is going to check your board paper, they will be getting it, and according to that only, it will be asked from us to check the paper. It is not like we will be checking according to our own wish. No, we cannot do that. So this marking scheme will be given. And same goes for the 5 marks also. So you can see question number 28, 1, 1, 1, 1, 2. Like this it is like. Then question number 31, it is 0.5 plus 0.5. Then for the point of question number 2, it is 1. Then third one, it is 0.5, 0.5. So like this, for each and every question, you will have such kind of the division. And in fact, step marking will also be there, like if you're writing the step. So in this question number 39, you can see 0.5, 0.5. Then question number 2 is 1 marks. Then question number 3 is 1 mark. Question number 4 is 1 marks. And then the last one is 0.5 marks. So this is called as a marking scheme, which you are not aware. They will be getting it then and there. All right.
Ma'am, what if you
Yeah,
don't
tell
write
me.
anything but you still draw the diagram? How many marks will they?
They have not asked you, but you are doing it, right?
Yes, ma'am.
See, it depends actually. It depends. Sometimes if it is not asked, you are drawing it neatly. In that case, they will give you the full marks. But if you are not drawing it neatly, and if it is not being asked also, in that case, instead of giving you anything, they will reduce your marks. So until unless you are not so perfect in making the diagram, don't draw it, children. If it is asked you to draw it, then definitely whether you know or no, Don't know, you have to draw it. But if you are not aware, they have not asked, then don't draw it. Okay?
Even if the points are correct and you draw the wrong diagram, they will cut marks.
Yes, they will reduce the marks. They will reduce the marks. They will not worry about the marking scheme in that case. So don't put this extra efforts. Until unless you are not very sure, don't do that. Okay, so coming to the differences between fission and fragmentation, the first point of the fission. In this method, how many parent cell will be formed? Yes, children, how many parents will be formed? Yes, children, how many will be formed during the fission time?
Two.
Two or more than two, actually. They're talking about fission, not about binary fission. So in the case of the fission, in this method, they will be dividing to form two identical daughter cell or more than that. And here, wait, here we should talk about the binary fission actually. So I did binary fission. Whereas in the fragmentation, in this method, the parent organism divides into two or more fragments. So in binary, it will be only two, and whereas in the fragmentation, two or more fragments will be developed, and each part develops into a new individual organism. So there it is two, whereas here it is the more than two will be formed. Then the next difference, it can take place in unicellular, it can take place in unicellular as well as multicellular. So both the kind of the organism will have this binary fission, multicellular organism. Whereas here in the fragmentation, it takes place only in multicellular organism. It takes place in multicellular organism only. Then the last point, example here, amoeba, paramecium. So both of them, what they will be doing, they'll be undergoing the binary fission, whereas here the example is a spirogyra. Fine, children. So these are the differences between the binary fission and fragmentation. So note this down.
Ma'am,
Yeah,
in
tell
which
me.
multicellular organism does binary fission take place?
In the
In which like a multicellular organism does binary fission take place?
hydra.
Right. Okay. Okay, ma'am.
In the hydra, you will have the budding also, you will have the regeneration also.
Ma'am, but then hydra also reproduces by fragmentation, right?
Correct. Yes. So in one individual, more than one process can happen.
Yes, ma'am. So, but then we wrote it as a difference, right? Like in binary fission, it happens for both unicellular and multicellular.
Yeah, but I have written here as a amoeba and the paramecium as an example. Okay. So in that case, you are talking only about the unicellular, not about the multicellular.
Okay, ma'am.
Done, children, with this page? Sumukh, Dhriti, Shubhansh,
Yes.
Puriya,
Yes, ma'am.
Yes, ma'am.
Arav,
Yes, ma'am.
Siddharth, Meher,
Yes, ma'am.
Yes.
Sudhamshu.
Done, ma'am.
Okay. So the next difference is what we're learning about is the
I'm
fragmentation.
writing.
Okay, okay.
Completed, ma'am.
Okay. So the next one are the differences between the fragmentation and regeneration. So fragmentation, it occurs in the multicellular it occurs in multicellular organisms with simple body. So in them, what happened? The body of these organisms is not complex. It is very simple, like as in Spirogyra algae. So they have a simple body organization. So they will be occurring in them. Whereas in the regeneration, it occurs in the complex or fully differentiated body. So regeneration occurs in fully differentiated multicellular organism with the complex body. So they will have the complex body, whereas in the fragmentation, they will have the simple body organization. Children, is just 1 minute. 1 minute. Just give me 2 minutes, okay? Let me see what happens. The next step is the next point.
Ma'am, can you want to show the points?
Done?
No, ma'am. No, 1 minute.
Okay.
Yes, ma'am.
The second point in this, an organism breaks into pieces and each piece develops into new individual. Whereas in regeneration, if organism breaks into pieces, they may or they may not pieces. Each piece may or may not develop into new individual. Like in the case of the lizard, what happened? Only the tail, only the body, from the body, the tail will regenerate. From the tail, the body does not regenerate. So that is why we say that in the regeneration process, maybe the each cut part will completely generate or maybe not. Then third point, here in the case of the fragmentation, they do not have specialized cells. So no specialized cells
Ma'am,
are,
here
yeah.
we written develop into new lizards.
Oh, sorry. Into new individuals, it should be. Yeah.
That was so random.
So actually, what is happening right now? They are crying outside like anything, and I'm not able to focus. So I am half present, half absent today because of them. So here in regeneration, what happens? Specialized cells will be there. What is the name of the specialized cells present in the regeneration process, or they are used in the regeneration process? A particular name I told you.
Regenerative cells or something like.
Very good. Regenerative cells only.
Oh, I don't know, ma'am. You gave some other alternative name also.
Specialized cell, regenerative cells, or totipotent cell.
Yes, ma'am. That one.
So any one name is like okay. Any one name can be written, but you should know all the three, because if they ask you any one particular term, so you should be able to mention that. So they will be proliferating and then they will be forming the mass of cells. So then this mass of cells, what do? They will differentiate. So the cells from the mass differentiate to form different cell types and tissues. So here in them, they have the regenerative cells, totipotent cells, or specialized cells, and they will be forming the mass. And this mass, they will be differentiating into the different cell types or the tissues type. So that is about the difference between the fragmentation and regeneration. So note this down.
Budding also uses regenerative cells, right?
What, Kashmi?
In budding, they
Yeah,
also
in the
regenerative cells.
no, in budding we do not have the regenerative cells.
Even in the textbook, ma'am.
Where in the textbook? Which textbook is given there?
In NCRT only, ma'am, in budding.
Okay, I'll just check it. But generally, the budding process which is happening, it is because of the karyokinases and cytokinases.
They
They do
said
not.
here in Hydra uses regenerative
Yes.
cells for reproduction.
Yes, in Hydra it is there. In the Hydra it will be there, but not in the case of the yeast. Hydra will have, but not the yeast. Done, children, with this page?
Yes, ma'am.
Yes.
Okay, go for the next one then.
Ma'am, once show the previous page, ma'am, please.
Oh.
Okay, ma'am, completed.
Hmm. It children, I'll just share this notes, okay, to you under the resources. Okay, I'll just end the class
Okay,
for
ma'am.
time being because